The central dogma describes the flow of information in a cell. DNA goes to RNA, which goes to a protein that makes a trait. Each strand of DNA has multiple genes, and each codes for a different protein. First, DNA is transcribed into RNA. In this process, the genetic material on a strand of DNA is copied onto a molecule called mRNA, which can carry the information to other parts of the cell. Three bases on an mRNA strand is a triplet that we call a codon. After that, mRNA is translated into amino acids, which make up proteins. This is the process of protein synthesis. Each strand of DNA has a 5' end and a 3' end, and consists of multiple bases that are paired together. These bases are adenine, thymine, cytosine, and guanine. When DNA is transcribed into RNA, the enzyme helicase has to unwind the DNA to be replicated, forming a replication bubble. After this, RNA polymerase is used to copy the DNA into mRNA, moving from the 5' end to the 3' end. The end result of this is a molecule of mRNA. Before going to a ribosome, mRNA must undergo gene splicing. When DNA is converted to mRNA, there are pieces of genetic material that are unusable. These are called introns, and they do not code for proteins but interrupt the sequence of genes. The pieces that code for proteins are called exons. To remove the introns, our cells use molecules called spliceosomes. A special type of spliceosome is called a SNRP. These sit on both ends of an intron and are used to remove it. To do this, the SNRPs pinch together until the intron breaks off, resulting in a usable strand of mRNA. There are three main types of RNA used in protein synthesis. The first is mRNA, or messenger RNA. It carries a copy of the DNA from the nucleus to the ribosomes, where protein synthesis takes place. The next is tRNA, or transfer RNA, which acts like a taxi and carries amino acids to the ribosomes. tRNA is shaped like a four-leaf clover that carries an anticodon at its base and an amino acid at the top. Last is rRNA, or ribosomal RNA, which assembles amino acids into protein chains. Unlike the last two, rRNA has a loose and messy shape. tRNA is involved in translation, which is a key part of the central dogma. At the bottom of a tRNA molecule is an anticodon that can bind to an mRNA codon, and at the top is an amino acid. Scientists use a genetic code chart to translate mRNA codons into amino acids. Each codon represents a specific amino acid that can be used to create a protein. For example, the code CUU could translate to the LEU amino acid. The ribosome is the site of protein synthesis. It is made up of two subunits, a large and a small. This can be visualized as two hamburger buns. Attached ribosomes are located on the rough endoplasmic reticulum, while free-floating ribosomes are located in the cytosol. Attached ribosomes make proteins that will be used outside of the cell, and free-floating ribosomes make proteins that will be used within the cell. The first step of protein synthesis at the ribosome is the mRNA binding to the small ribosomal subunit. In the analogy, the mRNA would be the meat inside of the hamburger. From there, the tRNA comes in carrying a specific amino acid. The anticodon on the bottom of the tRNA will bind with the codon that's located on the mRNA. This binding happens at what we call the A site. The A site is where the tRNA first enters the ribosome. Then, tRNA will go to the P site. The P site is where peptide bonds form between amino acids. tRNA then makes its way to the E site, where it will exit the ribosome. It will then go and retrieve another amino acid to repeat the process. As the amino acid grows, the ribosome will eventually reach a stop codon on the mRNA. At this point, water will enter the system and a process called hydrolysis will take place. Hydrolysis will break the polypeptides down and dismantle the system. To summarize, DNA will unwind and be copied into mRNA. From there, mRNA undergoes gene splicing. Then, mRNA leaves the nucleus to find a ribosome. tRNA will bring the correct amino acid to the ribosome, and then it will bind to the mRNA, releasing the amino acid. The amino acids will form peptide bonds, and then hydrolysis will take place, resulting in a newly formed protein.